Welcome to today's refresher lecture. I'm Dr. Leslie Blankenship-Williams, and today's refresher lecture is going to cover the first part of a complicated process known as aerobic respiration. Before we get into the nitty-gritty of aerobic respiration, we first need to ask, what's the point? In other words, why are we even covering this very complicated process? And clearly, it must be important. It's so important that many organisms on this planet use aerobic respiration in their every second life. It's not even everyday life, it's every second life, including humans. So once we answer what's the point, then we'll actually get into the nitty-gritty. With the nitty-gritty, we'll do an overview of the complex metabolic pathway that must take place. We'll talk about the importance of a special class of molecules called coenzymes and what their role is. And then finally, we'll look at the first two stages of aerobic respiration. Okay, so what's the point? In other words, why are we even covering aerobic respiration? In order to understand what's the point, we first need to look at the little economy that is the cell. You may recall from a previous refresher lecture that ATP is the cell currency. It is the molecule that is used to pay for cellular work. When you pay with ATP, you actually break ATP apart into ADP plus a phosphate group. And once that happens, that money is spent. So if you want to run the activities or the work of a cell, you're going to have to regenerate that money. Just like any economy, you got to spend money, but you got to make that money back if you want to keep spending it. So the real question isn't how we spend the ATP, it's how do we make it back? In other words, how do we regenerate that ATP? This is a question that every organism must address, or they won't be able to live. Interestingly, there's many strategies that can be used to regenerate ATP, including photosynthesis, chemosynthesis, anaerobic respiration, and even fermentation. But by far, the most common strategy used is going to be a strategy known as aerobic respiration. Now, before we move on any further, I want to talk about the word respiration. When I say respiration, a lot of people think breathing, respire, breathe in, breathe out, respiration. And of course, associated with that is the fact that when you breathe in, you're drawing in oxygen into your lungs and therefore into your body. And when you breathe out, you expel carbon dioxide. And we call that respiration. So respiration is bringing oxygen in and putting carbon dioxide out. For a whole human, that would be probably what you would think of when you say respiration. Is something else going on at the cellular level that is similar? And the answer is yes. Cellular respiration is indeed bringing in oxygen and putting out carbon dioxide at the cellular level. So at this point, now that we've linked what happens when we breathe with what's going on in the cell, let's move on and actually look at the overall metabolic pathway, that is aerobic respiration. Aerobic respiration starts with some type of food molecule. Glucose is the most commonly used, although it doesn't have to be glucose. It could be a fat, or it could even be a protein. But let's use glucose because that is the most commonly used substrate for aerobic respiration. Now, the word aerobic means oxygen. So in aerobic respiration, it's actually breaking down glucose with the presence of oxygen. So we take a glucose molecule and we combust it with 6 atmospheric oxygen molecules. Over many steps, what we end up with at the end is going to be 6 carbon dioxide molecules and actually 6 waters. So do you guys see the oxygen and the carbon dioxide in this chemical equation? We have the 6 oxygens that are required, and out comes the 6 carbon dioxides. This indeed is exactly why we need to breathe. We need to breathe so we can bring oxygen into the cell so that we can break down glucose. And we pump out CO2 because that's the waste product. Now you might look at this equation and say, and what's the end game? Why are we doing this? And it's not that we actually want to make carbon dioxide or even water. It's the fact that this is a catabolic reaction because we start with a larger glucose molecule and we break it down into smaller building blocks. In that process, we release energy, and it is the energy that is important. It's not the carbon dioxide or the water, it's the energy that we're trying to capture. So what's the point? The point is to somehow harness that energy released from the catabolism of glucose to power going from ADP plus a phosphate group back to ATP. So again, don't lose sight of the point. The point is to somehow break glucose down, get that energy out, and use that energy to basically reattach that phosphate group on ADP and regenerate our money so that the cell can keep doing work. So let's take another look at that metabolic pathway. Something to note about aerobic respiration. It can't happen all at once. You may recall in the previous picture, we had multiple arrows going. That means that we don't just do it all in one step. It's not as if glucose just goes, breaks down, and you have carbon dioxide. If we did, we would still release energy, but that energy would be released as heat, and we can't capture heat as a usable energy source. So instead, what needs to happen is we need to break down glucose a little bit at a time. You start with your glucose, you break off a hydrogen, you break off another hydrogen, you break off another hydrogen. Breaking glucose down a little bit at a time is going to maximize our ability to capture that energy and use it. So this patience in breaking it down a little bit at a time is going to reward us with getting the maximum amount of ATP out of one glucose molecule. And indeed, if oxygen is used in this catabolism or combustion of glucose, we can get up to 38 ATP molecules per glucose. So another look at what we're doing here. What we have here is we're going to take that glucose molecule. As you might notice, we have 6 carbons, 1, 2, 3, 4, 5, and 6 carbons. You can see them numbered right on the structure. And we have a bunch of hydrogens. Also, the energy of glucose is stored up in those carbon-carbon and carbon-hydrogen bonds. Every time we break off a hydrogen or we break a carbon-carbon bond and we replace it with an oxygen atom, a little bit of energy is released. So that's what we're going to do. We're going to start with glucose, and little bit by little bit, we're going to strip away the hydrogens and replace them with an oxygen. Break the carbon-carbon bond and plug in an oxygen. And that process is going to release the energy. So again, glucose. Of course, we need a source of oxygen. So here we go, our 6 oxygen atoms. We're going to use multiple chemical steps to get there, but eventually, what we're going to end up with is 6 carbon dioxides. For a moment, take a look at the 6 carbon dioxides, and you'll notice, the carbon dioxides are no longer attached to each other. In other words, we have no carbon-carbon chemical bonds, and we don't have any carbon-hydrogen bonds either. Instead, all of those chemical bonds have been replaced with oxygen, hence the carbon dioxide. And the hydrogens get moved to water. So if done correctly, we'll get 38 ATP out of that process. Okay, here's the tricky part. As we break down glucose one by one piece by one piece, we can't actually transfer that energy immediately into making ATP. In other words, we can't say, here's a glucose molecule, let's break off a hydrogen, and that little energy burst is going to be directly transferred to pushing that phosphate group back onto ADP and making ATP. It does work like that in a few instances, but mostly it doesn't. Instead, that energy that's released when a hydrogen breaks off needs to be temporarily stored somewhere. And that energy store is provided by coenzymes. So that's where these coenzymes come in. A service that they provide is temporarily harnessing that energy from the catabolism of glucose. And the goal will be to cash them in at a later time. A good analogy for coenzymes is like casino chips. When you go to a casino and you play, assuming you win big, you end up with a bunch of casino chips. Now, you can't go to 7-Eleven or to the grocery store and spend those casino chips as they are. It's not real money yet, but it represents real money. So casino chips are like the coenzymes. They represent energy, or money, if you will. They just need to be cashed in before we can get usable ATP. Let's take another look at what coenzymes are. This structure right here actually shows you one of the more common coenzymes called NAD. It's actually a nucleotide, but that's a really big, complicated chemical structure. So now that you've seen it, it's going to go away. And instead, we're going to use a simpler diagram to represent NAD. What I'm going to use is what looks like almost a little block with two divots in it. Those divots are going to be used to capture the energy. What does the energy come in? It comes in actually the form of electrons. So when we have glucose and we break off a hydrogen off that glucose, we have a hydrogen ion and 2 electrons, and we need to put them somewhere. They can't just go off into the cell. So we actually tag them onto this coenzyme called NAD+. Now, once we gain those electrons and that hydrogen ion, we have NADH, which is really the reduced form of NAD+. So you'll notice in this picture here, we have what's called a reversible chemical reaction. In other words, the NAD can go between the two forms, the oxidized form and the reduced form. Remember, a reduced molecule is one that gains electrons. An oxidized form is one that loses the electrons. So before we move any further, let's kind of devise a strategy for how NAD+ is going to work. Assume for a moment that you start with glucose and you break off a hydrogen atom. That hydrogen and its electrons need to go somewhere. So when we start here with this NAD+, we're going to go ahead and add this hydrogen and the 2 electrons, which are represented by blue balls, and we're going to put them right over here and make NADH. So the H represents the hydrogen, and we have these 2 little balls which are each supposed to represent the electrons. Okay, so this right here is the form that represents money. These are your casino chips. This is this form that you have that temporary energy store. So as we go through the first couple stages of aerobic respiration, we're going to be getting a lot of these reduced NADHs. And the point, of course, is that you're collecting casino chips, as it were, and shoving them in your pocket. At a later date, we're going to cash in those casino chips and actually get a lot of ATP. So, with that in mind, let's go through the stages of aerobic respiration. Aerobic respiration is commonly broken down into 3 stages: glycolysis, citric acid cycle, sometimes also called the Krebs cycle, and then a final stage called oxidative phosphorylation. Today, we're only going to look at the first 2 stages, glycolysis and the citric acid cycle. These 2 stages collectively will combust glucose into carbon dioxide plus water. In other words, it is at these 2 stages that we strip all the hydrogens off glucose and break all those carbon-carbon bonds. And you might think, well, isn't that everything? That seems like the entire chemical reaction. And it is almost. Here's the problem. As we go directly from glucose all the way down to carbon dioxide, those 2 processes, glycolysis and the citric acid cycle, only give us 4 ATP molecules. In other words, we do get a little bit of usable money right away, but it's nowhere near the 38 ATP we were promised at the beginning of this lecture. $4 does not equal $38. So clearly, there's a huge gap that we need to fill somehow. So where did the energy go if we broke glucose down correctly? And the answer is in coenzymes. Most of the energy that is captured in the stages known as glycolysis and citric acid cycle is not captured as ATP, it is captured in the form of 2 coenzymes, NADH and FADH2. So with that in mind, let's go through and start looking at the stage. We're going to start with glycolysis. And as you look at this picture, you might think to yourself, whoa, lots going on. And indeed, there is a lot going on. We start with glucose, and through multiple steps, we start breaking it down and stripping away those hydrogen. Instead of looking at all the details, let's do some basic bookkeeping. What we're really interested in is, what do we start with, what do we end with, and what did we get out of it? So those are going to be the questions we'll ask. So let's go with what we started with. We start with a single glucose molecule as shown here. At the end of this metabolic pathway, we end up with 2 pyruvate molecules as shown here. Each pyruvate is a 3-carbon molecule. So what did we get from this stage? Not much. As it turns out, we end up getting a net of 2 ATP. If you go through all the steps, you'll see that initially you actually have to pay into ATP. We call that the ATP investment stage. And the rule is, it takes money to make money. So it's true here as well. You actually have to pay in before you get a return on your investment. So we paid into ATP, and then at the end we get out for ATP. So if I were to give you $2 as part of my investment, and you were to give me $4 back, my net profit would actually be $2. 2 invested, 4 return equals net profit is $2. So the same is true here. We pay into ATP, we get out for ATP, and the net profit is actually 2 ATP. We also got a little bit of extra energy in the form of our coenzymes or casino chips. In addition to the 2 ATP, we also got 2 NADH as well. So at the end of glycolysis, we're left with 2 pyruvates, 2 ATP that we can go and spend right away, and 2 NADH that need to be saved in our pocket for a later step. So now let's take a look at what happens to the pyruvates. The pyruvates actually kind of go through what is called a prep step before entering the citric acid cycle. The pyruvate at this point is going to go from the cytoplasm into the mitochondria. And as it does that, it sheds one of its carbons. So you might notice here in this picture that pyruvate actually has 3 carbons, 1, 2, and 3. This top carbon is going to be shed off as it enters that membrane. And it sheds it off as one of the carbon dioxides. As it's shedding off that carbon dioxide, it needs a place to put the extra electrons. So where does it put them? Onto NAD+, of course, thus reducing it into NADH. So the end product here of this fairly complicated step is something called acetyl-CoA, which is actually a 2-carbon molecule. So what did we get out of this prep step? Well, we've gone from pyruvate to acetyl-CoA, we've released a carbon dioxide, and we've gained an NADH. So now let's take a look at what happens with the citric acid cycle. The citric acid cycle is another set of really complicated reactions. But once again, let's just do the bookkeeping. What did we really get out of doing this part of the procedure? And really what we're doing is we're breaking the rest of our original glucose molecule, now in the form of acetyl-CoA, down into carbon dioxide. So this is basically taking the remnant, which is acetyl-CoA, and just finish breaking everything off and making carbon dioxide. So what do we get out of it? Let's count. We get, if you look at this picture, you'll see 3 NADHs, 1 FADH2, which is another type of coenzyme, very similar, and then 1 ATP. So our net output, or our net gain, is going to be 3 NADH, 1 FADH2, and 1 usable ATP. Now, it's important to understand that per glucose molecule, this actually goes around twice. Because remember, in glycolysis, we started with 1 glucose, but we broke it down into 2 pyruvates. Each pyruvate is going to become its own acetyl-CoA, and then 1 acetyl-CoA will go through the cycle, and then the other acetyl-CoA will go through the cycle. So per glucose, our net gain is really double what I just said. In other words, really what we're looking at is collecting 6 NADH, 2 FADH2s, and 2 ATPs. Let's summarize what we've talked about. So let's put all of those steps together. We started with glucose, we broke it down into pyruvate, 2 pyruvates. What did we get? we got 2 ATP, and we got 2 NADH. Then we took that pyruvate and we broke it down a little bit further into acetyl-CoA plus carbon dioxide. The carbon dioxide is waste, and it basically goes away. This is what you breathe out. So there goes the carbon dioxide. So what did we get that we actually care about? 2 NADH. Okay, but now here's the big payout. We're going to take those 2 acetyl-CoAs and we're going to finish breaking them down into 4 carbon dioxide molecules. Once again, the carbon dioxide is waste. It's what you breathe out when you respire. So it goes away out of the lungs. But what did we get? We got 6 NADH, 2 FADH2s, and 2 ATPs. So when we add all of this together, through glycolysis, the PrEP, and the citric acid cycle, so far, we're done with our organic molecule. We have no more organic molecule to deal with, but we get 4 ATP, 2 FADH2s, and 10 NADHs. So, as I mentioned, we only get 4 usable dollars right now, but we have a whole pocket full of casino chips. And the very last step, oxidative phosphorylation, is about cashing in those casino chips and getting the big jackpot payout of ATP. But that will be for the next refresher lecture. Thank you very much.